Welcome to The Explainer. Today, we are diving into a complete step-by-step blueprint for modernizing your analytical workflows. We're talking about the AI-assisted scientific analysis pipeline. Now, let's get one thing straight right out of the gate. We are absolutely not framing AI as some kind of magic wand that just does the science for you. No way. Instead, I want you to think of it as an incredibly advanced co-pilot for scientific discovery. It's a highly structured methodology that uses large language models, or LLMs, to basically supercharge your work, taking you all the way from raw data collection to a beautiful final visualization. But before we get into the nuts and bolts of how this works, let me ask you a question that I'm sure hits pretty close to home. Honestly, how much time do you waste writing boilerplate code instead of doing actual science? We all know that awful, overwhelming dread of manual formatting, wrestling with weird syntax errors, and configuring basic statistical scripts when literally all you want to do is test your hypothesis. Well, this explainer gives you a systematic way to shift your focus back to the real research. We're going to break down exactly how to let the AI handle that repetitive execution so you can stay focused on the broader scientific vision. So here is our roadmap for the journey. We'll be tracking how to take your raw data and turn it into publication-ready visualizations across 5 phases. First, building the data foundation. Second, AI-powered parameters and methods. Third, vibe coding the analysis. Fourth, validating the results. And finally, visualizing for publication. Let's get right into it. All right, phase 1, building the data foundation. Look, before any AI intervention can even happen, you need a rock-solid, incredibly clean base. Preparing for an AI pipeline involves this mundane but absolutely critical reality. If you feed an LLM messy data, you are going to get messy science back. It's that simple. First up, you have to gather all your experimental parameters and raw measurements, making sure to meticulously include your units and provenance. Then, systematically classify the data type. Are we talking time series, tabular, image, signal, text? Documenting this explicitly is a must because it literally dictates the applicable methods we'll use later on. And finally, document your metadata. Note the data size, dimensions, completeness, and seriously, make a special note of any missing values or outliers. This pristine foundation is the prerequisite for the whole shebang. Moving right along to phase 2, AI-powered parameters and methods. Building out your parameter intelligence follows a pretty simple 4-step sequence. First, you toss your full list of parameter names into an LLM, like Claude or GPT-4. Second, you prompt it to evaluate their analytical significance. Third, and I cannot stress this enough, this is absolutely crucial. You, as the human expert, have to review that output and cross-check it against your own domain knowledge. Remember, the AI is just a tool, not the final authority here. Then, fourth, you extract that refined, curated parameter set for your downstream analysis. What you're left with is this highly relevant list of parameters, complete with a documented rationale for exactly why each one was either kept or tossed out. So, the secret sauce here is knowing exactly how to ask the AI for help with your methodology. Once you've got your data type, size, format, and that newly refined parameter set dialed in, you hit it with this exact prompt. What analytical methods are applicable to this data set and parameters? It's simple, but it is a powerful, precise question that perfectly sets up the AI to uncover the best research directions based strictly on your data's unique profile. And the response you get back isn't just some random list of statistical tests. Because you asked the right question, the LLM gives you a ranked guide. It actually maps statistical, machine learning, or signal processing methods directly to your potential research directions or hypotheses. You're essentially using the AI as a high-powered brainstorming partner to find the most mathematically sound pathways to answer your scientific questions. All right, on to phase 3, which is where things get really fun. Vibe coding the analysis. So we've reached the execution phase. Now, if you're a traditional researcher, the phrase vibe coding might sound a little out there, but it's actually an amazing concept. Vibe coding just means using natural language prompts to rapidly generate and run boilerplate code using AI-assisted tools. So instead of burning hours writing out the exact syntax for a statistical method from scratch, you literally just describe what you want in plain, everyday English. The AI then takes your intention and translates it into executable code. It's a total game changer. To actually pull this off, the pipeline recommends a sort of modular toolkit. A great option is Cursor, which is an AI-native code editor that gives you inline completions. You can pair that up with GitHub Copilot for context-aware code suggestions as you're working. And obviously having Claude or ChatGPT open in another window is just invaluable for code explanation and for debugging when you inevitably hit a snag. When these tools work together, they accelerate your coding process to a degree that is hard to overstate. So here is what that fast-paced vibe coding sequence looks like in practice. First, open your tool and prompt it with the specific method name, your parameter list, and your data format. Second, let the tool auto-generate the boilerplate code for that method. Third, you run that newly generated code using the default parameters, just to grab your baseline results. And lastly, carefully log all your outputs and any error messages you run into. Now notice the huge step that's missing here. You didn't write the baseline code yourself. You just directed the AI to write it for you, which drastically cuts down your execution time.
Okay, phase 4, validating the results.
Moving from rapid generation over to validation, we need to slow the pace way down and apply some rigorous scrutiny. Your initial validation is all about comparing the AI-generated outputs against your original hypothesis. The next steps are pretty cut and dry here. If the results align with what you expected, awesome, you proceed straight to a deeper mathematical analysis. But if the results diverge, don't panic. You just revisit your earlier parameter selection or rethink your method choice. It is a built-in checkpoint just to make absolutely sure your AI copilot hasn't steered you off into the weeds.
So assuming things look good and you're proceeding, you enter the deep dive phase. This is where you iteratively refine your analytical approach. You might need to tune parameters, like adjusting thresholds, hyperparameters, or window sizes. You will very likely need to adjust covariance, adding or removing confounding variables, like when you're calculating an adjusted odds ratio. You might also explore stage-wise implementations, which means applying methods sequentially or as an ensemble. And you absolutely should be running sensitivity analyses to see how robust your results are to input variations. Make no mistake, this right here is the heavy lifting of real science. To keep you from losing your mind with all that complex refinement, you'll want to use a fine-tuning checklist. This maps your specific checks to actions and keeps track of their completion status. You systematically verify hypothesis alignment, tweak key parameters, control for those tricky confounders, run your multi-step approaches, and ultimately verify that your scientific robustness is up to strict peer review standards. Keeping a close eye on this ensures your AI-assisted workflow stays highly disciplined and grounded. And finally, we arrive at phase 5, visualizing for publication.
This is the final polish, where we prepare to actually share our validated findings with the rest of the scientific community. When you're picking your chart types and using those publication quality libraries like Matplotlly, Seaborn, Plotly, or ggplot2, you have got to stick to strict best practices. Always, always label your axes with proper units. Make sure you include the sample size n and your confidence intervals wherever they apply. Be sure to use colorblind accessible palettes so your data is actually readable by everyone. And lastly, save your visuals in high resolution, at least 300 dpi, so they look crisp and are ready for print. But listen, a pretty graph is never the finish line on its own. The ultimate goal of this entire AI-assisted pipeline is complete, flawless scientific reproducibility. You have to meticulously document your methodology, the exact parameters you fed in, and your interpretations in a final summary report. You want to annotate your plots with statistical significance markers and error bars, showing your key findings right alongside clear next steps. Yes, the AI helped you get here a lot faster, but the absolute transparency of your reporting is what makes it valid, trustworthy science.
Which leaves us with this final provocative thought for you to chew on today. When an AI pipeline steps in to handle all that tedious boilerplate, the repetitive parameter filtering, and the basic code execution, what happens to your role? It essentially challenges you to stop acting like a code monkey and step fully back into your true calling as a scientific visionary. If the AI is managing the execution, well, the only limit left on your research capacity is the quality of the questions you choose to ask. Thanks so much for joining me for this explainer.